Well, thank you, Barbara, for the introduction, and thank you for the invitation to speak to you this afternoon. So since this is a somewhat diverse audience in terms of background and depth of experience and thinking about specifically bringing genomics into oncology, my goal for this talk is to throw out some first principles just to make sure we're all on the same page about what we mean when we say precision medicine, and then go through some of the aspects that in our experience at an academic cancer center, some of the key components that need to be in place, where the challenges around implementing those components are, and then sort of touch on what will be delved into much more deeply by the other speakers, which is sort of clinical frameworks by which we can test this hypothesis that bringing genetics and other molecular information more deeply into our thinking in terms of clinical trials can have a substantial impact. Let's see. Okay, here we go. Okay, so let's start with some guiding principles, or our first principles of precision medicine. So the first of these is, and I'm starting relatively basic. I'm going to assume you all know this, so I'm just going to begin to kind of make sure we're all on the same page. The first is that, I don't know what I'm supposed to be pointing at to make this. Okay. Molecular pathways involved in tumor survival and progression are often activated by genetic alterations. And this is, I'm sure everybody knows this kind of at a gestalt, but actually if you go through several of the major cancer types and enumerate the, point this way? Okay. Enumerate the spectrum of mutations where if you see it, you can already think of something you might do in terms of either a clinical trial based on a targeted therapy that's available, or maybe steering away from a certain category of agent. The numbers are actually quite interesting. So these are 6, so lung adenoma, lung squamous, breast cancer, colorectal, melanoma, head and neck, and then down here we have in bar graph form ovarian cancer and glioblastoma. And what you can see, I won't walk you through all of these pie charts, but you can see that they're actually fairly full. And if you extract these, you get to the recognition that in several major tumor types, 40 to 60% harbor at least one genomic alteration that affects an actionable proliferation or survival mechanism. And so actionable here can either mean, in rare cases, it means that there's already an FDA-approved agent. In more cases, many more cases, it means that there's a clinical trial agent that either hits the target or the pathway. And equally commonly, it may mean that there is an alteration that would steer you away from using a particular category of agent. So we're using the term actionable broadly, but what the term has in common is seeing that alteration is not just for the cancer biologist to get excited about a pathway. It's actually something that may evoke in the back of a clinician's mind a decision or a different choice of care. So that's the first principle. The second principle is that anti-cancer agents targeting many oncogenic pathways have entered clinical trials. So this is actually, we are in a unique point in history. This is the first time where we can say that for the major cancer signaling pathways and survival pathways that have been studied in the lab for the past 3 or 4 decades, there is not just one, but multiple drugs targeting multiple components in those pathways. And so this is illustrated schematically. Here's the MAP kinase pathway where there are RAF, MEK, and ERK inhibitors, multiple RAF and MEK inhibitors, and then recently 1 or 2 ERK inhibitors have entered trials. Of course, the PI3 kinase pathway, multiple nodes here have, again, multiple drugs being developed against those target proteins. Of course, a whole slew of receptor tyrosine kinases. And of course, these are all pathways that undergo recurrent genetic alterations in various different cancer types. One can go down the line, the Notch, smoothen signaling, and other CDK inhibitors, CDK, cyclin-dependent kinases, et cetera. So we're sitting here in a unique point in time where the pathways that we have known about for a long time, now there is a whole repertoire of developmental drugs that can, in principle, intercept those pathways. So that's the second principle of precision medicine. And the third one is that we now have the technology to look for these alterations, thereby matching the right tumor to the right drug, at least in principle. So the genomic technologies have advanced to the point where it is possible to do robust genomic profiling in the clinical arena. And so the nature of that shift can be illustrated by comparing what we currently would do in most sort of standard, perhaps community settings or even academic settings, which is to think about the cancer type based on its known histology. Think about the most highly recurrent alterations in those cancer types, of which there are maybe one or two, and then think of a test or maybe a couple of tests to run that will tell us what drug to use. But what's become increasingly apparent is that there are, Bert Vogelstein describes there are mountains and there are hills. The mountains mean the very common genetic alterations that occur in cancer that we can all name, EGFR in lung cancer, KRAS in colon cancer, et cetera. But then there are hills, which are the much less common events to that specific cancer type, but they do occur. And in fact, the hills may even outnumber the mountains. So what that means is that we don't necessarily just want to read out one or two genetic alterations. We may want to read out an entire panel of alterations for which there might be a drug available. And the ability to do that has rushed forward. And in fact, the enabling technology that you all know about is massively parallel sequencing. This slide's already out of date because it says that Illumina an Illumina instrument can generate 300 gigabases per 8-day run, which is 100 times a single genome. This actually is now up to about 600 gigabases. So the power to generate massive amounts of data is here. And in fact, the turnaround time for generating such sequencing data is shrinking. The analysis tools that are needed are becoming more and more available. So basically, we are now in a situation where we have all the ingredients that are needed to practice precision medicine. We have the knowledge of tumor biology. There's a wide range of actionable alterations that occur in cancer. We have drugs that hit those alterations, and we have genomic technology that can be acted upon in the clinic to actually mix and match, and therefore test the big idea of precision medicine. So those are the first principles. And on top of those principles, one can now build an engine, if you will, that will, now this, what I'm putting up here is not meant to be something that would be done on every cancer patient, but certainly it's a set of capabilities that academic medical centers are building so that they can run the types of precision medicine trials that are most exciting. So it, of course, starts with the patient encounter. Very often, this is paired with a fresh biopsy, although that's not certainly mandatory. One can also take archival material, but certainly fresh biopsies can be interesting. A profile is done. The data is interpreted and returned to the oncologist so that a decision can be made. That decision can be about whether or not to enroll into a particular clinical trial, or it could even be about binning patients who are in a clinical trial into particular subgroups for which one or another drug might be chosen. One then monitors the response, and then even later, if a patient did respond and they progress, understanding mechanisms of resistance with yet another biopsy at that point. During the setting of clinical response, one can do even another biopsy to read out pharmacodynamics. So this engine and having it be in place is really what enables the possibility of the most exciting precision medicine trials, because it provides a basis for answering some of the major questions that are driving our attention in terms of new therapeutics. So for example, does genetic or molecular stratification identify patient subgroups that in fact do benefit from novel agents? Does the drug in fact inhibit the relevant oncogenic pathway? So doing biopsies here and profiling here can answer that question, because obviously if you didn't hit the target, you didn't really do the clinical experiment. What are the mechanisms of resistance to existing or emergent agents, and what combinations hold promise in various genetic subtypes to achieve more durable control? So one needs many of these components in place to deeply interrogate these questions. Okay, so now what I'm going to do is take this engine and sort of invert it and make a couple of points. So the first point is that although this is exciting, this is very hard. It's daunting even to the most advanced cancer centers. And in fact, an analogy was made at our institution just last week when we had a joint cancer meeting at the Broad Institute, where we invited Atul Gawande, who many of you know for his writings, his books, his New Yorker contributions. And so he came up with this very clever analogy, as he is known to do, where if you think about what is happening here, so I'm not quite sure what I'm supposed to be pointing at to advance the, okay. So if you think about what's happening sort of at the top part, particularly when you think about profiling and the omics that's possible, you can think of that now in terms of the genomic information that's coming out of tumors as the superhighway. But the rest of it is really much more akin to a bike path. So he had this analogy of, you know, the genomic superhighway meets the bike path of clinical medicine. And that basically, maybe he'll publish a book on that someday, but it was a great, it packed the room. The title of the lecture packed the room. But it really was prescient, because what it brought to the forefront is when we think about the clinical trials that we're doing, they're not just about efficacy. They're not just about whether or not a new drug works. They're about this entire process, because it doesn't really exist in cancer centers. There are rapidly expanding endeavors to implement it, but even understanding how to do that and what works, what doesn't, is part of the entire clinical research enterprise in a way that I certainly hadn't appreciated before. Okay, so just briefly going through some of the key points and the challenges that exist here. Some of these will be, of course, well known to the clinical trialists in the room, but other subtleties are maybe more or less well known. So when one thinks about enrolling patients into a genomics-driven paradigm, the nature of the consent form actually becomes highly relevant, because what one has to do is consent the patient not only to undergo comprehensive genetic testing and share that data, which obviously has potential loss of privacy concerns. Actually, that issue was brought to bear recently in an article in the scientific literature showing that you can, in fact, if you're clever, discover the identity of patients whose genomic information has been sequenced and made publicly available. So this has to be included in consent forms, as does, and protocols need to be built that allow the return of genetic results, not just a single mutation here or there, but return of large-scale genetic results. This is a major area, an investigational area. It's not straightforward to write protocols that do that. So this is something that requires attention right off the bat and can take quite a while to finish. So when one thinks about the tissue and the sort of complexity of what needs to happen, you know, often when we think about multiple biopsies and biopsying not just the tumor, but also the matched blood or matched normal tissue, and having to take them down separate paths for characterization, you know, sample tracking, having a sophistication there that maybe is larger than what commonly exists in many settings, having rigorous approaches for quality control, and also making sure that the appropriate paired normal material is used, particularly when one is expanding beyond just a few genes, but to much larger panels or the exome scale. And so, sorry, what am I supposed to be pointing at? Oh, is that what it is? Oh, there we go. Okay. So then there's the question of the profiling itself, and I think this is obviously requires a lot of attention. What is the nature? What's the breadth of information that needs to be gleaned? And that really will depend heavily on what the drug is, whether it's a selective targeted drug or whether it's a kind of a multi-targeted drug, or it's a combination without sort of a clear-cut genetic context or what have you. But there are a variety of profiling approaches at one's disposal that start from being relatively simple, which is just genotyping for panels of mutation, to more targeted, all the way up to whole-exome RNA, and even epigenetic studies are coming in. So this clearly, this is going to be a major topic for consideration, which I think is a great opportunity, but also potentially challenging for cooperative group trials to think carefully, depending on the study, what's the nature of the profiling that's useful, and then where does the capability to do this profile need to come from? Can it be built sort of internally to clinical trial infrastructure, or does it need to be outsourced? So briefly, at our institution, we've put together a clinical study. It's a prospective study, which we've called CanSeq. And the idea is to prospectively perform whole-exome sequencing on cancer patients from these, starting with these 4 malignancies at the Dana-Farber, and then to not only generate the sequencing data, but to return the results to the clinical team. So this is, so we are into year 1. We're actually actively enrolling patients on this trial now. These are the cancer types that are part of the study. But just on the next slide, you can see, by way of example, the nature of the complexity. So of course we're consenting patients at the Dana-Farber. There's a separate consent form that they receive, and clinical research coordinators approach the patients and the oncologists in the disease center. The specimens are then coordinated for enrolled patients in the Department of Pathology. They actually then move across town. They're sent across town to the Broad Institute, where the exome sequencing is done. By the way, an aliquot is saved for CLIA validation of actionable events that might be of interest. But the Broad will shortly have an arm that allows CLIA exome sequencing, so that issue won't be as important. But then there's a whole area of research around how you decide from the exome what is in fact actionable. Once that decision is made, the exome data goes to a committee that reviews the data, and ultimately a report is returned to the treating oncologist. And so this then brings up the next issue in the chain, which is interpreting the data. So it's actually, at this point, it's not hard to generate the data. The interpretation of the data, however, is much more challenging, and this can be illustrated. So this is a slide that's going to be very familiar to many people, which is the comparison of the cost of sequencing data generation to Moore's law, which I'm sure many of you have seen this slide. But an aspect of the slide that maybe is not as familiar to people is when one considers the data points that are generated. So the data points per patient as a result of implementing a genome type sequencing data, compared to what the typical constellation of data points is that we consider as clinicians. So if you think about sort of the number of data points per patient in sort of the typical clinical workup over the past decade or so, it's sort of in the low dozens of independent values that we really care deeply about following. But once you bring in even just the lowest level of genetics, just a targeted mutation panels, you get up easily over 100. And then once you bring in exome, larger gene panels, or certainly exome level, it shoots way up. So the number of data points per patient is skyrocketing, and this needs to be solved. So what this means is that there need to be clinical data interpretation algorithms that are capable of addressing this. And so I tell you, I'm really failing this test of advancing the slides today. Okay. So one of the members of our team has tackled this issue by developing a web-based, heuristic-based model for interpreting the alteration landscape. So the concept is that out of an exome or a transcriptome comes a list of mutations. It goes through an algorithm that's built based on these components, which I won't walk through in great detail. By the way, the nerds in the audience will recognize that the name PHIAL comes from the Lord of the Rings. It is the phial of Galadriel, which was presented to Frodo at a very dark time in his mission, with the quote, "May it be a light to you in dark places when all other lights have gone out." So, for the nerds, it's lyrical to us, because this is what we're doing for cancer patients, hopefully, who are out of options. We're doing an exome or genome and giving them options that they might not otherwise have had. So, we were very proud of that. Okay, but the point is that one has to have a framework for taking the superhighway of information and converting it into something that an oncologist can use. And so another component of this is to have an approach to tiering the evidence, obviously based on FDA-approved or standard therapies, and then various phases in each tiers that we can bin various alterations into to make it potentially useful in the clinical decision process or the clinical trial enrollment process. Okay, so then there's a web-based tool that one can then scroll through and consider the data. And this has been quite useful to members of the team because they can sort of scroll into various content and sort of see the set of alterations that are seen, to the extent to which they might be actionable. And so this is actually the team of faculty with various areas of expertise in oncology or genetics who are beginning now to evaluate the exomes that have come through and then help make decisions about what should be reported back to the treating clinician. So in the interest of time, I'll skip this. So then, of course, one needs to communicate this, and there's a whole area of research around how best to make reports available. And this is an example of an earlier report that's come out for exome level data at Dana-Farber. And then finally, the question is, does the information provided impact clinical decision making? And here's where we actually really do feel that there is a lot of exciting information in these exomes. One of the interesting questions will be comparing the value of an exome or a transcriptome with more targeted data, but certainly there is rich information, and it's already beginning to have impact on clinical trial enrollments. Here's one brief example of a patient with lung cancer, an unusual history. He had a history of breast cancer, and then he developed a lung adenocarcinoma, rapidly recurred after surgery, progressed rapidly through standard therapy, and tested negative for all of the standard alterations that are being now tested in lung cancer. The exome data revealed several alterations that, after going through the interpretation and curation, were deemed plausibly actionable, some of which are shown here, and the way in which the annotation was done is shown here. And the result was that a clinical decision was made to enroll a patient on a clinical trial specifically based on this sort of atypical KRAS mutation. So, and I think you'll hear many more examples of this from David Solit and Vince Miller going forward. So finally, I'm just going to close by pointing out that the bottom of this was really actually the entree into where the cooperative groups sit in many ways, because now the question is, when you put this infrastructure into place with all the attendant challenges around data generation and data interpretation, do we improve care at the end of the day? And so there's going to be, hopefully, lots of discussion about various categories of clinical trials, which many of them fall into this category of either genotype to phenotype or phenotype to genotype. So genotype to phenotype is the idea of you take a genetic set of genetic information, use that and the phenotype will be clinical response, so we think we can use this information to pick drugs for clinical response. Whereas the phenotype might be that you saw a clinical response, but you don't know why, and then you use the genetics to sort out was there a genetic basis, and David Solit will talk more about that. And there are a variety of other types of studies, decision impact, how often do you switch your care based on what was done, and then comparisons of various platforms. So we'll talk more about the concept of a drug-centered or a basket approach, where one starts with a drug, such as a RAF inhibitor, and goes across one or multiple types of genetic indications and tests the benefit of that. And then there's more of a, I'm not sure what the right term is, but the battle-like approach, where one generates a genomic profile of which there may be many mutations. One has multiple drugs at one's disposal. And then one basically tests the idea of the targeted group, where the genetics was used to guide the choice of therapy, to comparing to, for example, an empiric group where that wasn't done, and then a series of endpoints can be evaluated. So I'll stop there, but hopefully this lays out sort of first principles of precision medicine and various challenges that need to be addressed, and the other speakers will unpack more.